The Human Genome Project has been enormously successful. We've spent billions of dollars to figure out what are the piece parts of the genome. What are the genes? Where are they? What do these piece parts do? What we have only begun to do is, how do you take all those piece parts and in a given individual say, this is what it means?
The Clarity Challenge is an effort to take children who have a serious and heritable disorder, sequence their DNA, and seek to find clues as to the basis for that child's disease. We decided to attract the best practices from around the world and seek to understand how the most skillful teams do the work of sequencing and interpreting human exomes and genomes while making it safer for patients and more easily understandable for doctors.
For the purposes of the challenge, we chose 3 families who had what we believed to be a genetic condition, but for which previous research and clinical testing had been unsuccessful. Family number 1 has a boy named Adam Foy, who's 11 years old, who has a condition we call centronuclear myopathy. Family number 2 was a family with an unusual cardiac condition. There was a brother and a sister, and each had had one affected child. Sadly, one of these children, Liam, had been born with a very severe defect in his heart and resulted in his passing when he was only a few days old. Family 3 had yet a different condition we call Nemelin myopathy. In this case, there was an affected boy and 2 unaffected parents. He had a mutation in a gene that hadn't been identified yet or that nobody had had the ability to look for. The material that was made available to the contestants was the gene sequences and the clinical information.
Many different groups from around the world entered the Clarity Challenge. There were government labs, there were academic research groups, a number of companies who are in the field of DNA diagnostics or who write software for DNA diagnostics, and there were a few enterprising small groups of biologists.
By having these teams be willing to literally in a matter of weeks do what used to be a multi-year study, we demonstrated that it was possible, and I think what we've done is accelerate the reality of the clinical practice of clinical genomics so that it can be practiced now rather than in 5 years or in a decade.
Out of the 23 complete entries, there were 8 outstanding entries, and of those, there were 3 finalists. The first finalist had a very good protocol for asking the patient ahead of time what they'd like to hear back, so-called setting patient preferences. The potential problems with whole genome sequencing is that you may find more than what you're just looking for. In addition to finding the cause of the patient's weakness or heart rhythm disturbance, you may discover something else about them or about their future. The question is, how do you deal with that information, and does the patient and their family really want that information back, or are they prepared to receive it? The second finalist actually identified all the potential disease genes correctly, which none of the other entries had done. The winner had the best combination of bioinformatic analysis as well as mining of the medical literature to put together the big list of potential mutations, and a really excellent report that was readable by a physician who was not an expert in the disease and interpreted to the family. The results for Family 1 for Adam Foy were really quite exciting. A number of groups identified mutations in a gene that encodes for a protein we call titin, and the titin protein acts as a spring to help keep the muscle together as it contracts and expands. We're very fortunate in being able to study the titin gene in zebrafish. One of the approaches that we hope to be taking in the next few years will be to use zebrafish with a titin mutation as the basis to screen for drugs that might allow the muscle to form more normally and function better. For family number 2, who had baby Liam, what we found is that Liam carried a mutation in a gene called TRPM4 that's responsible for controlling the electrical currents in his heart. Knowing this now, we can identify family members who might carry this gene and who might be at risk for a heart rhythm disturbance, and there may be drugs in the future that specifically target it to treat their arrhythmias.
I could not help but be gratified and amazed that so many teams were willing to invest their time and efforts to actually compete. They really wanted to move the science forward.
Prior to the contest, there weren't clear standards on how to interpret the measurements made in DNA sequencing. As a result of the contest, I expect that this will accelerate the responsible use of genomic information for diagnostic purposes.
Just by showing that it's actually doable, that dozens of teams were able to actually perform this task, I think sends the message out. The time is now right. The time is now right to do genomic medicine.